Hello everyone. Today we will know about the plasma membrane or cell membrane. So what is the cell membrane or the plasma membrane? First of all cell must have a plasma membrane or the cell membrane. Look at this structure. It is a cell. What is the cell membrane? That means the outermost boundary or the outermost structure of the cell is called as the cell membrane or the plasma membrane. So first of all, in the 1950, the electron microscope was discovered. So after the electron microscope discovery, then only the discovery of the plasma membrane was done. And the first discovery of the plasma membrane was done in the cell that is called as the RBC, or the red blood cell. That means in the RBC, the first analysis of the cell membrane was done. So now look at this structure. This is the cell membrane structure. So if I zoom this portion, it looks like this. That means actually the plasma membrane is not a single layer structure. It is a two layer structure. This is the one layer. This is the second layer. Or it is called as the leaflet. Leaflet. That means two leaflet. This is the one leaflet. This is the second leaflet, or it is called as the one layer or second layer. So always that's why the cell membrane is double membrane structure. So now come to the structure of the plasma membrane. What is the constituents of the plasma membrane? So in the plasma membrane, mainly the lipid and protein are present. Okay. So this structure is called as the lipid. This is the one structure. This is the one lipid. This is another lipid. This is third lipid. So now come to the what is the structure of the actual lipid. The one lipid structure is looked like this. That means it has two segments. One is called as the head, or it is called as the head part, and it is called as the tail part. So head part always the hydrophilic. What is the hydrophilic? The structure which always wants to interact with the water molecule is called as the hydrophilic. So what is the tail? Tail is the hydrophobic structure. Hydrophobic structure. That means it is the same structure in which both hydrophobic and the hydrophilic structure are present. So that's why the lipid is called as the amphipathic molecule. Amphipathic molecule. So what is the amphipathic molecule? That means any structure which contains both hydrophilic as well as hydrophobic structure, that is called as the amphipathic molecule. So now in the lipid, in the head part, actually in the head part, it contains glycerol along with phosphate. So this is the head part. And in the tail part, this is the one tail, this is the another tail. So what is this tail part? These are called as the fatty acid. Fatty acid. So now what is the fatty acid? Fatty acid is simply the chain of the hydrocarbons, and at the last part the COOH group is attached. So this is called as the fatty acid. The fatty acid can be two types. One is called as the saturated fatty acid, and another is the unsaturated fatty acid. So what is the saturated fatty acid? That means in the fatty acid, if all the bonds are the single bond, then it is called as the saturated fatty acid. So this type of structure of the fatty acid is called as the saturated fatty acid. So now look at what is the unsaturated fatty acid. That means in the fatty acid, if at least one double bond is present, or more than one double bond is present, then it is called as the unsaturated fatty acid. Okay, so now you know what is the saturated fatty acid, what is the unsaturated fatty acid. So look at the structure of the, so what is the lipid? The lipid is the at the head part, the glycerol and the phosphate. Look at the structure. This is the glycerol structure, this is the phosphate structure, and this is the tail. So tail means it is the only the fatty acid. So lipid means glycerol, phosphate plus fatty acid. So one is the saturated fatty acid, another is the unsaturated fatty acid. The saturated fatty acid, it actually present like this, that means in a straight line structure. But in the unsaturated fatty acid, because of the presence of the double bond structure, it bends somewhere, it bends, or it is called as a kink, K-I-N-K, kink. So what is the kink? That means simply the bending. That means if the saturated fatty acid is present in the lipid, then it looks like this. If one unsaturated fatty acid is present, then it looks like this, or it is designated as this. So always normally in the membrane, the lipid contains one unsaturated fatty acid. So that's why if we look at the book, then it looks like this. That means one tail is bending like this. Here looks like this. One is simple straight, another is bending. Why it is bending? It is called as a kink. That means in that area a double bond structure is present and the whole fatty acid is unsaturated fatty acid. Okay, so plasma membrane contains protein plus lipid. So actually which type of the lipid is present? So normally, or in the plasma membrane, the lipid is phospholipid. Phospholipid. What is the phospholipid? That means the lipid in which the head part contains a phosphate group. Here the PO4, a phosphate group is present. That's why it is called as a phospholipid. Okay. So always in the plasma membrane, the lipid is the phospholipid, and another lipid is present. That is called as the sterol. So what is the sterol? Sterol is a subgroup of steroid. Subgroup of steroid, or the group, one of the group of the steroid is called as the sterol. So different types of the sterol are present in different organisms. For example, in the animal, the sterol is present, that is called as the cholesterol. In the plant, the sterol is present in the plant, is called as the phytosterol. In the fungi, the sterol is called as the mycosterol. So in different organisms, different types of the sterols are present. So always in the plasma membrane structure, the lipid is present, and which type of the lipid? One is the phospholipid, another is the sterol. So two types of the lipid are present in the plasma membrane. So now come to the protein. Along with the lipid, some proteins are present here. In between that, some proteins are present. So this is all about the structure of the plasma membrane. So now come to the models of plasma membrane. So what is the models of the plasma membrane? That means the plasma membrane, or what are the sequence of the discovery of the plasma membrane. The first model was discovered and it was called as lipid and lipid bilayer model. It was discovered in 1925. Okay, and it was discovered by Gotter and Grendel. Gotter and Grendel. So what is the lipid and the lipid bilayer structure? This structure is called as the lipid and lipid bilayer model, which was discovered in 1925 by Gotter and the Grendel. What is the lipid and the lipid bilayer? So Gotter and the Grendel, they speculated at that time that only the lipid are present in the plasma membrane. This is the one layer, that is the lipid layer. This is the another lipid layer. That means there is no protein, there is no carbohydrate, only the lipid are present according to that model, which is wrong. But at that time, actually it was a first model. So that's why they thought that it may contains only the lipid layer. That's why it is called the lipid and the lipid bilayer. That means it is a bilayer structure, and the always means in all areas lipid are only present. That means no protein, no carbohydrate is present in that plasma membrane. So the second model is called as sandwich model. Sandwich model. It was discovered in 1935 by Danieli and Devson. So what is the sandwich model? It is the sandwich model. This type of the structure is called as the sandwich model, which was discovered in 1935. So according to this model, here at the outer side the protein is present. Here it is the one protein layer, here it is the one protein layer, and then the inside the lipid layer is present. That means it is called as the sandwich model. That means the lipids are sandwiched by the two protein layers. That means inside the lipid is present, and this lipid layer is sandwiched by protein. That means the outer here the protein, here it is the also the outer protein. That means two proteins, this sandwich the lipid structure. So this is the second model. The third model is called as unit membrane model. Unit membrane model, and it was discovered in 1953. And I have already told that the first electron microscope was discovered in 1950. So, and it was discovered by 1953. That means it is the first model which was discovered after the discovery of the electron microscope. So that is called the unit membrane model, and it was discovered by Robertson. Okay. So what is the unit membrane model? According to that, here the protein layer, this is the unit membrane model. So according to that, the protein layer, it contains protein and lipid according to this model, and the protein is asymmetric. That means not same in the both layer. That means at the outer layer, look at this structure, it is a thick, and at the inner layer, this is the thin structure. The thin layer of the protein is present, and inside it the lipid is present according to this model. So that is called the unit membrane model. So the fourth one is most important, that is called as the fluid mosaic model. Fluid mosaic model, and it was discovered in 1972 by Singer and Nicholson. Okay. So please note that the fluid mosaic model, which was discovered in 1972, it is the most accepted theory. It is the most accepted theory of the plasma membrane. That means even nowadays we also accept the fluid mosaic model. It is the latest model and it was accepted only about the plasma membrane structure. So that's why if it in the exam, if it will be asked that which is the most accepted theory or the which is the accepted theory or which is the latest theory about the plasma membrane model, and the answer will be the fluid mosaic model, and it was discovered by Singer and the Nicholson, and this is the structure of the fluid mosaic model. That means nowadays whatever we know about the plasma membrane, it is according to the fluid mosaic model. So now come to the extensive study about the fluid mosaic model. So this is the fluid mosaic model. According to that, the plasma membrane contains lipid plus protein plus carbohydrate. Okay. So what type of the lipid are present? One is called as the phospholipid. Another is called as the sterol. So now look at the structure. This is the one layer of the one leaflet. This is the another leaflet. So this structure is called as the lipid structure, and this orange color structure, it is called as the phospholipid. Okay. So the yellow color structure or this structure or here this structure is called as the sterol. Here it is called as the cholesterol. Why it is called as the cholesterol? Because it is the animal cell. This is the animal cell. Why? Because in the animal, the sterol is called as the cholesterol. If it is the plant, the sterol is the phytosterol. If it is the fungi, the sterol is called as the mycosterol. So one is the phospholipid and the sterol. So if in the exam it will be asked that what are the types of the lipid present in the plasma membrane, and it is called as the simply phospholipid and the sterol. And what is the function of the sterol? It helps in the fluidity of the plasma membrane. Fluidity of plasma membrane. Okay. So this is the phospholipid. Phospholipid, that means this type of the structure is called as the phospholipid. One is called as the head part, and it is the hydrophilic, and it is the tail part, and it is the hydrophobic. Now, what do you think that why the head part is called as the hydrophilic, or why the head part is hydrophilic and why the tail part is hydrophobic? Look at the structure. Let's say this is the cell and this is the membrane of the structure. So that means it looks like this. That means here one is the head part, here at the outer side the head part is present, and then the inside it or the middle like this, here the hydrophobic group or the hydrophobic tail group is present. But why? Always in the cell, let's say this is the one cell, this is the another cell, this is the another cell, this is called as the extracellular matrix. Extracellular matrix. And the inside it, or the inside of the cell, is called as the cytosol. So always in the extracellular matrix of the cytosol, the watery condition the watery solution is present. That means the cytosol is simply the watery condition. So that's why the inside the water molecule, let's say water molecule, not water, the watery solution or the hydrophilic solution is present. At the outside also, this is called the extracellular matrix. Here also the hydrophilic or the watery molecules are present. So that's why the head part is hydrophilic, so that it can interact with the water molecule. So always the hydrophilic, they want to interact with the water molecule. And what is the hydrophobic? They always want to away from the water molecule. So that's why at the middle part, here the hydrophobic or the tail part is present. At the outer side or the inner side, always the head part is present, so that it can interact with the water molecule. In the middle or the core part, it doesn't want to interact with the water molecules. So that's why they always want to away from the water molecules. That's why they are present at the inside of the cell. So this is the phospholipid. This is the sterol, which helps in the fluidity of the plasma membrane. So another type of the lipid is also present, that is called as the glycolipid. Glycolipid. So now So one is the lipid part. So in the lipid part, one is the phospholipid, another is the sterol, another is the glycolipid. What is the glycolipid? We will know after some time. Okay, this is the lipid part. So in the plasma membrane, lipid plus protein is also present. So what is the protein? So here the blue color structure is called as the protein. Here it is the one protein, here another protein, here the another protein. So the proteins are two types. One is called as the peripheral, another is called as the integral. The peripheral is also called as the extrinsic protein, or the integral protein is also called as the intrinsic protein. So what is the extrinsic, what is the intrinsic protein? So extrinsic, now, or the peripheral, as the name suggests, that means it is only present at the periphery. Look at the structure of the protein. This is present only at the outer area of the lipid, and this is called as the extrinsic of the peripheral. So what is the integral? That means it must have gone to the core of the plasma membrane. So look at the structure. It already buried in the core of the plasma membrane. That's why it is the integral or the intrinsic protein, and this is called as the peripheral or extrinsic protein. Okay, so because the peripheral protein, they are present only at the outer side, it binds the lipid membrane very loosely bound. And integral protein, because it is already buried to the middle of the bilayer, it is strongly bound to the lipid bilayer. Okay, so this is the structure of the lipid. Another lipid, okay, one is the peripheral, another is integral. Some protein like this is also a protein. It already present at the inside and the outside. That means it already buried whole part of the lipid bilayer, and this type of the protein is called as the transmembrane protein. Transmembrane protein. So normally three types of the protein are present. One is the peripheral, which is present at the just outside at the periphery of the lipid bilayer. One is the integral, that means it is already present near the core of the plasma membrane. Another is the transmembrane, which buried along the plasma membrane, that is called the transmembrane protein. So another structure is called as the carbohydrate. Carbohydrate. So the carbohydrate look like this type of the protein, this type of the carbohydrate. Carbohydrate are simply two types. One is called as the glycoprotein, another is the glycolipid, which is already I have told. So what is the glycoprotein, what is the glycolipid? So always carbohydrate always present at the outer side, that means either this side or this side. It is not present at the middle of the plasma membrane. And carbohydrate is only present by binding either the protein or the lipid. So glyco means carbohydrate, and the protein means protein. Here it is the glyco means carbohydrate or carb, the lipid. So that means some carbohydrate binds to protein and it is called as a glycoprotein, and some carbohydrate are present by binding with the lipid, and this is called as the glycolipid structure. Look at this structure, this is called as the glycoprotein. That means this is a protein. To this protein a carbohydrate group is attached, and this carbohydrate is called as the glycoprotein. And some protein are present or bound to the lipid structure, and that is called the glycolipid structure. So in summary, in the plasma membrane, lipid plus protein plus carbohydrate are present. Then the lipid, one is the phospholipid and the sterol, they are present in the plasma membrane. Another is the glycolipid is present, which is present at the outer side by binding with the carbohydrate. Another is the protein. There are three types of the protein. One is the peripheral protein, which is present at the periphery. Another is the integral, which is buried in the integral part of the plasma membrane. Another is the transmembrane, that means it already buried to the whole plasma membrane structure, that is called the transmembrane protein structure. Another is called the carbohydrate. Carbohydrate is present only at the outer side by binding with the lipid and the protein. If it is bind to the protein, then it is called as the glycoprotein. If it is bound to the lipid, then it is called as the glycolipid. So this is all about the plasma membrane structure. So now in some different cell, different types of the composition of the lipid and the protein and the carbohydrate present. In the exam always asked what are the constituents or the composition of the lipid and the protein and the carbohydrate present in the RBC. Why it is asked in the RBC? Because the first discovery of the plasma membrane was done in the RBC. So in the RBC, the 52% lipid are present, 52% protein are present, 40% lipid are present, and 10% carbohydrate are present. Okay, so this is the structure of the lipid bilayer. So one is called as the, why it is called the fluid mosaic model. The structure is called as the fluid mosaic model. One is fluid, another is the mosaic. Mosaic means like the tiles. The mosaic, the fluid is due to the lipid structure, and the mosaic is due to the protein plus sterol. What is the mosaic? That means like the tile structure. If in the house, then the tile house, the tiles are present at the out like this. So this is called the mosaic. Here also, the lipid and the protein and the sterol are present like the mosaic model. That's why it is called the mosaic. And what it is called the fluid, and the fluid is due to the lipid. Why it is called the fluid? That means the plasma membrane is not very strong or not very hard or not very rigid. It is very, very soft. Okay. So unlike the cell wall is very hard or the very rigid, which is present at the plant cell. That means in the plant cell, this is the plant cell, this is the plasma membrane, and outer side of the plasma membrane another layer is present, and it is called as the cell wall. The cell wall is very hard, but the plasma membrane is not very hard. It is a fluid like structure or the very soft. Why it is called as a fluid like structure? Because it always rotates around each other. That means it is not fixed. That means let us say this is the lipid bilayer structure. It is always not fixed. It is always in a movement like structure. That means always it is moving. The structure is always moving, and that's why it is called the fluid structure. So why or the how the fluid or the how the lipid is moved? Lipid moves by two methods. One is called as the flip flop, another is called as the lateral movement. If movement of the lipid, there are two types of the movement of the lipid. One is called as the flip flop, another is the lateral movement. What is the flip flop? Look at the structure. If this lipid it comes to this layer, and this lipid comes to this layer, this is called as the flip flop movement. That means from the one leaflet to another leaflet, or from one layer to another layer, then it is called as a flip flop, or simply it is a vertical movement. That means from one leaflet to another leaflet. What is the lateral movement? That means if this lipid it comes to here, and here it comes to here, then only it is called as the lateral movement or the horizontal movement. So always in the plasma membrane, it is not fixed. They move always around each other. So that's why the lateral movement of the protein, lateral movement of the lipid, it is responsible for the fluidity of the plasma membrane, along with the cholesterol. Previously we have already studied about the sterol. That means the sterol it maintains the fluidity of the plasma membrane. For the lateral movement of the lipid, it is responsible for the fluidity of the plasma membrane. So along like the lipid, the protein, let us say in the inside some protein are present. The protein are also moved, but only in the lateral movement. That means the protein can move only laterally. That means in one leaflet only, but it cannot move like the flip flop. That means the protein cannot go from the one layer to another layer, but the lipid can. So lipid can move from the flip flop or the lateral movement. That means lipid can move horizontally as well as vertically, but the protein can be moved by the laterally. That means in same leaflet or the same layer, it can go or it can move, but in the horizontally, in the vertically, they cannot move. So this is the movement of the protein in the lipid. So now come to the function of the plasma membrane. What is the function of the plasma membrane? So what is the function of plasma membrane? One of the function is called as the cell growth. Cell growth. Cell growth means, let us say this is a one cell, it grows like this. Why? Because the plasma membrane is fluid, not hard. Let us say this is, if it is the hard like cell wall, then the growth of the cell cannot be done. So that's why one of the function of the plasma membrane is the cell growth, another is cell division. Cell division. Cell division means one cell divides to two cells, or the mitosis and the meiosis you will know about after some time. So the mitosis, the meiosis, two types of the cell division is present in the cell, and the cell division is also due to the plasma membrane. Another is endocytosis. Endocytosis. What is the endocytosis? Let us say this is the cell. Some molecule is present outside the cell. It enters into the cell and it is called as the endocytosis, or the engulfment of the cell, or engulfment of the molecule by the cell is called as the endocytosis, and it is also done by the plasma membrane because the plasma membrane is fluid or the soft. And the fourth one is intercellular junction, formation of intercellular junction. What is the intercellular junction? Let us say this is the one cell, this is the another cell, this is the another cell. So here the cell junction, the cell interaction is also helped by the plasma membrane. This is the function of the plasma membrane. Now come to the transport of the plasma membrane. Transport across plasma membrane. So what is the plasma membrane which helps in the transport across? So always the plasma membrane, it is a gatekeeper, it acts as a gatekeeper. That means, let us say it is a cell, it is a gatekeeper. That means it permits the molecule by seeing what is the composition of this molecule. This is called the gatekeeper. That means it selectively allows some molecule. So that's why the plasma membrane is also called as the selectively permeable membrane. Permeable membrane. Or it is also called as the plasma membrane is also called as the semi-permeable membrane. Semi-permeable membrane. That means this membrane or the plasma membrane structure, it doesn't allow to all the molecules. it selectively allows some molecule, that's why it is called as the selectively permeable membrane or the semi-permeable membrane. So what are the different types of the transport across the plasma membrane? There are the two types of the transport are present. One is called as the passive transport, another is the active transport. So what is the passive transport? The passive transport means that any molecule which can transport without use of the energy, or the active transport, it transports to the cell or the outside the cell by using the energy. Now come to the passive transport. The passive transport means the molecule transports along the concentration gradient. What is the along concentration gradient? That means from the higher concentration to lower concentration. Look at this structure. Let's say this is the outer surface, outer layer. This is the inner layer. What is the outer layer? That means let's say this is the cell. It is the inner, it is the outer. Let's say this structure, if it is zoomed, it looks like this. That means it is the outer layer, it is the inner layer. Look at this molecule. This molecule is present higher concentration at the outer side and the lower concentration in the lower side. That means here the two molecules are present in the inside, and the 1, 2, 3, 4, 5, 6, more than 2 molecules are present at the outer side. So that's why this molecule can directly pass from the higher concentration to the lower concentration, and this is called as the along the concentration gradient. Okay, so the passive transport, it is two types of the passive transport. One is called as the simple diffusion, another is the facilitated diffusion. What is the simple diffusion? The simple diffusion means the gas, for example, the oxygen and the carbon dioxide, they can pass easily through the plasma membrane along the concentration gradient. Let's say this is the cell. In the inside, let's say the carbon dioxide is more. Suppose at the outside the carbon dioxide is low concentration. That means let's suppose the 5 molecules of the carbohydrate is present at the inside and 3 carbo CO2 molecule, they are present at the outside. That means in the inside more carbohydrate is present, more carbon dioxide is present. That means that now the carbon dioxide transports from the higher concentration to the lower concentration, or the along concentration gradient. That means from the inside to the outside. Let's suppose in the inside the oxygen is present in the lower concentration, at the outside the oxygen is present at the higher concentration at the outside. Now the oxygen comes from the outer to lower. That means molecule which is present higher concentration, and the movement is always from the higher to lower concentration. If it is present at the higher in the inner surface, then it transports from the inner to a higher. If it is present higher in the outer, then it transports from the higher to the from the outer to the lower, outer to the inner layer. So this is the simple diffusion. One is the gases, another is the non-polar molecule. For example, benzene. Benzene is a non-polar molecule. Why the non-polar molecule they can pass? Because the lipid layer is itself it is a non-polar or the hydrophobic. Always the lipid bilayer, the plasma membrane is hydrophobic. So that's why the hydrophobic molecule they can easily pass the layer, because it can means this is the hydrophobic molecule, here it is also the hydrophobic molecules. That's why they can mix each other and they can easily pass through the hydrophobic layer. But if it is a hydrophilic molecule, this is the hydrophilic molecule. The hydrophilic molecule they cannot pass through the hydrophobic. Why? Because the hydrophilic cannot interact with the hydrophobic molecule. Because the hydrophilic means those molecules which interact with the water molecule. But the hydrophobic means those molecules which always wants to away from the water molecule. That's why the hydrophilic cannot interact with the hydrophobic molecule. That's why the hydrophobic molecule cannot pass, but the hydrophobic molecule they can pass through the layer. So that's why gases, oxygen, carbon dioxide, any gases, and the non-polar molecule, that means the hydrophobic molecule, they can pass also the plasma membrane from the higher concentration to the lower concentration. Another most important thing, one is called as the diffusion. What is the diffusion? Diffusion is the transport of water molecule through the simple diffusion. Sorry, what is the not diffusion? That is the osmosis. Osmosis. What is the osmosis? The osmosis means the transport of the water molecule through the simple diffusion, that is called the osmosis. So that means the osmosis is the sub part of the simple diffusion. That means the simple diffusion, the gases can be passes from the higher concentration to the lower concentration. The non-polar molecule they can also passes from the higher concentration to the lower concentration, and the water molecule also can passes from the higher concentration to the lower concentration through the simple diffusion, and this process is called as the osmosis. So what is the osmosis? It is the movement of the water through the simple diffusion. Through the simple diffusion is called as the osmosis. Okay. So this is the simple diffusion. That means it always moves from the higher to lower concentration or the along the concentration gradient. That's why, because it transports from the higher to lower concentration, it does not need any energy. It does not need any energy. So another is the facilitated diffusion. So what is the facilitated diffusion? For example, some molecule, let's say protein. Let's say this molecule, it is the protein. Normally the protein is the hydrophilic molecule. Hydrophilic molecule. Let's say this protein has higher concentration at the outer side. Let's say this is the protein molecule, and it is the hydrophilic. Okay, it is present at the higher concentration at the outer side and the lower concentration higher concentration at the outer side, lower concentration at the inner side, but it does not pass through the membrane directly, because this is the hydrophilic molecule. It cannot interact with the hydrophobic core group, because the plasma membrane or the lipid is a hydrophobic group. But if the non-polar molecule, non-polar, for example the benzene, if it is present at the higher concentration at the outer side, lower at the inner side, it can directly pass through the layer, because the lipid layer is also the hydrophobic, the non-polar or the benzene is also the hydrophobic. So it can interact with each other and they can easily pass through the membrane. But now the protein is the hydrophilic, it does not, or it cannot pass through membrane directly. So that's why it needs some assistant, and this assistant is called as the carrier. That means it needs some carrier for the transport of the molecule from the higher to lower. So this is called as the facilitated diffusion. That means some facilitated needs for the transport of this molecule, and this is called as the carrier, or it is called as the carrier mediated facilitated diffusion. This is called as the carrier mediated facilitated diffusion. Okay. So now there are the two types of the transport. One is called as the simple transport, another is the facilitated diffusion, another is the simple diffusion. So what is the simple diffusion? So the gases, oxygen, carbon dioxide, non-polar molecule like the benzene along with the water. Water is the exception, that means it transports through the process that is called as the osmosis. So osmosis is simply the simple diffusion in which the molecule is only the water. That means the movement of the water through the simple diffusion is called as the osmosis, and they along with the facilitated diffusion. The facilitated means the hydrophilic molecule or some polar molecule, if it is present higher concentration, then it transports from the higher to lower concentration, but it is facilitated by some carrier, that is called as the carrier mediated facilitated diffusion. Okay. So this is the simple and the facilitated diffusion. So now come to three types of structure. One is called as the hypotonic, isotonic, hypertonic. Hypotonic means it is the inside or the inner side, this is the outside. Hypo means low, hyper means high. So in the solution, one is called as the solute, another is called as the solvent. When the solute means the hypotonic, isotonic, hypertonic solution, they are named according to the solute concentration. That means in the hypotonic solution, at the outer side more solute concentration, means low solute concentration is present. That means in the inside higher solute concentration is present, at the outer side lower solute concentration is present. In the outer low solute, so if the low solute is present at the outer side, that means more solvent. is present. In the inside, higher solute is present, that means low solvent is present. Isotonic means in the outer side and the inside all are same. This is called the ideal condition of the isotonic solution. This is the hypertonic solution. In the hypertonic solution, the outer condition contains more solute at the outer. That means low solvent is present at the outer. Now, in the exam, it will ask that if any cell, let us say this is the solution and this is called as the hypertonic solution. Hypertonic solution. In the exam, if it will ask that, let us say this is the one molecule or the one cell, if one cell is transported or is placed to the hypertonic solution, then what will be the condition of that cell? Then the answer will be shrink. Why? Let us say in this area. Now the cell is placed in the cell, inside the cell. This is the hypertonic solution. This solution, this is called as the hypersonic solution. In the hypersonic solution, the solute concentration, the solute is more and the solvent is less, like this. So that means in the inside, the solute will be low and the solvent will be high. So that means let us say the solvent is the water. The water molecule is present, this is the solvent, means water. So now that means the water molecule is present higher concentration in the inside and the lower concentration is the outer side. So that means it transports from the higher to lower. That means the water released from the higher to lower, that means from the inner to higher. So that means after the transport of the cell, the placing of the cell to the hypertonic solution, after the transferring the water molecule from the inner to higher, it will shrink like this. Why? Because after transporting the hypertonic solution, the solution of the water concentration is less at the outer side and the water concentration is more in the inside. That means water, they transport from the inner to lower, that means from the cell to the outer solution. So that is why it will be shrink. In the exam, if it will be asked that if the cell is placed to the hypertonic solution, it will shrink. So like that, if the cell is transported to the hypotonic solution, let us say this is the cell, this is the hypotonic solution. In the hypotonic solution, the solute concentration is less and the solvent is more. That means in the inside the solvent is less. The solvent means let us say water. Obviously in the cell the solvent is the water. That means in the hypotonic condition, the water molecule concentration higher in the outer side and then lower in the inside. So from the higher to lower, that means the water they transports from the higher solution, from the outer solution inside to the inside the cell. That means from this solution it transports to the cell, so that after transferring so many water molecule, the cell will be burst. If the molecule, if the many water molecule they are transported inside the cell, it will be burst. So if the cell is transported from to the hypertonic solution, the cell will be shrink. If it is transported to the hypotonic solution, it will be burst. Okay. So now come to, this is the passive transport. Okay, passive means one is the simple diffusion, another is the facilitated diffusion. Simple means the gases and the non-polar and the water molecule, they can pass easily from the higher to lower concentration or along the concentration gradient. Facilitated means the polar molecule transport from the higher to lower concentration. But what if, let us say, this is the cell. Always inside the cell, the sodium concentration is very low, and the outer side the sodium concentration is high. And always inside the cell, in the any cell, the potassium concentration is high, and in the outer surface, the sodium concentration the sodium concentration is low. Okay. So that means in the simple diffusion or the facilitated diffusion, it always the sodium transports from the higher to lower, that means from the outer to the lower, from the outer to the inside, the sodium transports. But the potassium through the passive transport always transports from the inside to the outer side. Okay. But in sometimes the potassium transports from the outer to lower, or the sodium transports from the lower inside to the outer side. And this type of the transport is called as the active transport. Active transport. So that means here the active transport means transport of the solute or the transport of the molecule against concentration gradient. Against concentration gradient means from the lower concentration to the higher concentration. So because against concentration gradient, it needs some use of the energy. Look at this structure. In this structure, let us say this is the outer, this is the inner layer. In the outer layer, low concentration is present, and the inside higher concentration is present. So if in the passive transport, it can easily pass from the inner to the outer, but it cannot pass from the outer to the lower. but if it wants to transport from the outer to the lower, or from the lower concentration to the higher concentration, it needs some energy. Here the energy is the ATP. Don't confuse between the energy and the ATP. That means if it will ask that in the active transport the energy is needed or ATP is needed, always mention the energy is needed. Why? Because ATP is one of the type of the energy. That means we use ATP as a form of the energy. For example, some bacteria like halobacteria, they use light as a source of energy here, but still it is called as the active transport because of the investment of the because of the use of the energy, not the ATP. We use ATP as a source of energy. Halobacteria, they use light as a source of energy. So that means in the active transport, or the transport which moves against the concentration gradient, that means from the lower to higher concentration, the energy is needed. Either it may be in form of the ATP or it may be in the form of the light. Okay. So one of the most example is the sodium potassium pump. Sodium potassium pump. Okay. Now this is the sodium potassium pump. This is the sodium potassium pump. This is the extracellular fluid. That means it is the outer side. This is the cytoplasm. That means it is the inner side. So I have already told that always inside the sodium concentration is lower and the potassium is higher. And the outer side the sodium concentration is higher and the potassium concentration is lower. So that means in the passive transport, potassium can easily pass from the inner to outer, and the sodium can easily pass from the outer to the inside. But if sodium wants to come from inner to outer, that means it is the against, or the potassium it wants from the outer to inner, it transports, this type of the transport is called as the active transport, and this type of the is called as the sodium potassium pump. Look at this structure. This is called as the, this type of the structure, it is called as the sodium potassium pump. And here 3 sodium, 1, 2, 3 sodium molecules are transported from the inner to the outer, and like that 2 potassium molecule transports from the outer to the inner, and this is called as the active transport. And here the ATP is needed as a source of the energy. So the sodium potassium pump, it is the example of the active transport through which 3 sodium and the 2 potassium are transported at the same time through this pump, and this pump is called as the sodium potassium pump. Okay. So this is the all about the plasmam structure and the transport of the across the plasma membrane. Okay. If you like it, then please subscribe. Thank you.